Good afternoon. I'm Alfonso Caramazza, the co-director of the Mind, Brain, and Behavior Interfaculty Initiative. I'd like to welcome you to the second of talks by Randy Gallistel, who is professor of psychology and director of cognitive science at Rutgers University. MBB, as many of you know, is a university-wide effort to promote interactions at the boundaries or the borders of disciplines interested in the mind, brain, and behavior. And the MBB Distinguished Lectures that we're holding today embody, in many respects, the broad spirits of this initiative. The lectures engage with themes that cross traditional disciplinary boundaries among the mind, brain, and behavior. As said yesterday, for those who were here, but for people who were not here yesterday, past MBB Distinguished Lectures have included luminaries such as Jean-Pierre Changeux, Antonio Damasio, Kay Redfield Jameson, Noam Chomsky, Nicholas Humphrey, William Newsom, Martha McClintock, Stanislas Dehaene, Daniel Kahneman, Dan Dennett, Michael Gazzaniga, Patricia Kuhl, and Uta and Chris Frith. And as many of you heard yesterday from Liz Spelke's introduction to the series of talks by Randy Gallistel, he's a most prolific contributor to many diverse areas of the cognitive neurosciences. His research interests span from genetic approaches to the neurobiological mechanisms of memory to the cognitive mechanisms in spatial, temporal, and numerical learning and reasoning. He has received many honors and recognitions for his work, including election to the National Academy of Sciences and the American Academy of Arts and Sciences. Yesterday, he gave a fascinating and provocative, really provocative, lecture entitled "It's the Neuron, How the Brain Really Works." It generated a lively discussion with the person who was commenting, John Listman, and I can say that the discussion degenerated and the lively debate continued unabated into the evening with other MBB colleagues until around 11 o'clock. We were all tired except for Randy, who managed to win every argument and enjoyed it. Today, the lecture is entitled Perception of Probability. I'm sure it will be as fascinating as yesterday's lecture, and perhaps as provocative. And the talk will be followed by a brief commentary by Rick Bourne, who's professor of neurobiology at Harvard Medical School. Please join me to welcome you again, Randy Gallistel.
Thank you very much, Alfonso. It sounds like you can hear me clearly now, right? Okay, good. I have to remember to start my time. Okay. Then, there we go. So this is collaborative research that I'll be reporting. As you can see, there's a number of collaborators. It's both experimental and modeling, with more emphasis on the modeling than the experimental part of it. As you see from the title, it's about the perception of probability. And the last time I gave this lecture, someone I know very well asked very reasonably, they said, why do you talk about the perception of probability? So I thought, rather than, I'm not quite sure what she was suggesting, but I understand the kind of thrust. I think I understand the thrust of the question, because I think we don't ordinarily think of perception of probability as being something that's perceived, at least in the same sense in which we perceive the shape of an object or a color. But in fact, I realize that I've spent a good deal of my research career working on what I sometimes call the psychophysics of the abstract, or the psychophysics of the abstract dimensions of experience, such as distance, duration, rate, and probability. And I've worked on both these perceptions in both humans and in animals. And all of them have the property that they lack qualia. That is, we become aware of these things. These dimensions of experience are thoroughly experienced in rats and in mice, so there's nothing particularly human about this. In fact, I think these are really the most basic dimensions of experience, the most primitive and the most important behaviorally. But none of these dimensions has qualia. One of the experiments we did some while back in collaboration with Frank Durgan was people's perception of how far they have walked under conditions where there was no sensory input from the outside. They were wearing blinders, they were wearing earphones, they had to count backwards by twos so they couldn't count their steps, and they had a rope that they hung onto so they didn't wander into the obstacles in the corridor, and we had them walk some meters down this very long corridor in an athletic building, and then we said, okay, now walk back until you think you're back where you started. We ran ourselves as subjects. It's a long tradition in psychophysical work. We also, of course, ran others. It's sort of indescribable how this works. I mean, you walk along, and after a while, you just feel you've gone the right distance, and so you stop. But there's no sensory content to the experience, nor would you expect there to be. Well, the same thing applies to probabilities. At least I think. We routinely become aware of the probabilities of different kinds of events in our environment, but there is no sensory aspect to this. There's no qualia to a probability. But it has an extremely important effect on our behavior. But there's almost no work on the computational modeling of how it is we extract from our ongoing experience from a series of events over time an estimate of the probability. And that's what this is about. It's built on an experiment, an unbelievably simple experiment that we ran. Ye Lu and Monica Krishan ran different parts of it at different times in my lab. The subjects, as in so many cognitive psychology experiments these days, are seated in front of a computer display, and they have this slider that they can move with the mouse, just the way I've positioned the cursor on it, to indicate their current estimate of the proportion of green circles in this box of rings. So the subjects are drawing from an urn full of rings but instead of marbles, but the same idea. And some of the rings in the urn are green and some are red. And every time they click next, they get to see another circle which emerges and then retreats back into the urn. So it's very clear that this is drawing with replacement from the urn, right? And their task is to estimate what they think the hidden Bernoulli parameter is. That is, what they think the proportion of green circles is in the box. Not the proportion they've seen, but the proportion that they think obtains in the box. Now, of course, this is based on, in some way, on the proportion that they've seen. So they're invited after every trial, but not required, to move the slider to indicate their current perception of that hidden aspect of the generative process. When they move the slider, the proportion of red and green dots, they're indicated here by black and gray dots, in this, there are 1,000 dots in this square. And as they move the slider, the proportion changes in the square, so that this gives visual feedback. We thought this would be important initially, but all the subjects said basically after a little practice, they never looked at the box. And when we ran ourselves, that was what we did too. You just basically didn't pay any attention to the box. You had a percept, you moved a slider to indicate your percept. An important thing was that the subjects were told at the outset that the urn would be silently replaced or might be silently replaced from time to time, and the new urn would have a different proportion of green circles. So it was a stepwise non-stationary Bernoulli process that they were looking at, And whenever they thought there had been such a change, they clicked on this button here on the screen that said the box has changed. Now, after we ran the first 5 subjects, Monica came to me one day and she said, you know, Randy, when I debriefed the subjects afterwards, most of the subjects say that on occasion they clicked on the box has changed, and then after they saw more circles, they thought it really hadn't changed. They had second thoughts. And I said, whoa, that is not a minor observation. This is, bears quite strongly on the kind of model you might entertain about how their percept is generated. And so we need to get data on that. So we added yet another button that said, I take that back. When they took it back, so they would click on this if they said there was a change, and then after a while they thought, you know, I don't think there really was a change back there. They didn't go back and adjust the slider setting retroactively, right? They just said, you know, I've had a second thought. I don't think there was a change. We didn't say anything about how often they changed or how big. In actual fact, the steps were pretty large, in most cases, fairly easily detectable, at least in principle. They were extremely unpredictable. That is, it was a geometric process that determined this, essentially a simulation of the exponential. And there was a 0.005, 1 chance in 200, that there would be a change after any given trial. This is a maximally random way of distributing the changes. It means that there can be and were as long as 400 or 500 trials in which there was no change in the box. But it also meant that there can be and were occasions in which the box only lasted 1 trial. So, of course, it's completely hopeless for the subject to estimate the probability under those circumstances. There's no way the subject would ever know that such a thing had happened. But because we have access to the computer, we know that such a thing actually did happen. There's a backstory to this. While we were running this, I kept saying to Monica, I said, look, I don't generally work with humans. I don't follow the judgment and decisions literature where all this work is done. This is an absurdly simple experiment. It's hard for me to believe that someone hasn't done this in the past. It's your job to find out if anyone has. So we asked Danny Kahneman and other friends of ours who work in this area, Gretchen Chapman and so on. They were unable to, they couldn't say no, that they knew of this. So we naively concluded maybe it hadn't actually been done. Then we sent it out for review, and oh God, the reviewers jumped all over us and they're claiming we hadn't cited X and Y and Z. So we sorted through that, and eventually it was done once before in a pre-computer age as a PhD dissertation by a man named Robinson back in operations research in 1964, using a lever instead of a slider, and a dial and flashing lights, so very different methodology. It was unbelievably gratifying that he got exactly the same results that we did. So that was very encouraging about the robustness of the results. And here's what the results look like. This is you trust me, this truly is a representative session. The subjects saw 1,000 circles in the course of one session. You think, oh my God, 1,000 circles. It only takes about 25 minutes. You really click through these circles pretty fast. After a while, like most psychophysics, it's very boring. We ran every subject for 10 sessions. So we have 10,000 circles and 10,000 slider settings for each of 10 subjects. That's 100,000. Psychophysics has always been sort of big data, at least by psychology's standards. What you see here is absolutely characteristic of every session in every subject. And the first thing you notice is, well, the solid line is the slider setting, the subject slider setting. The dashed line is the true hidden probability. And the first thing you say is that the subject, it's not bad. He's pretty close to the true probability most of the time. But what's very striking about this is that the subject is not changing the slider often for many trials at a time. Here we have more than 100 trials when the subject didn't move the slider. Now, So the subjects move the sliders intermittently instead of on every trial. That's already surprising because in a Bernoulli process, if you think, for example, the p is 0.5, then every datum that you observe is as far from your estimate as it's possible to be, right? Because it's either 1 or 0. And on the usual kind of model of how you estimate parameters in this kind of process, you should update after every trial. And that means your estimate is getting jerked around by the data all the time. I hope this is at least sort of obvious. So you would expect little perturbations in the slider on every trial, and you don't see it in any subject in any session. So that's one striking fact. But the other striking fact you see right here, which is that although often the slider doesn't move by much at all, in fact, not at all, there are occasions, usually driven by the input, in which the slider will move, or the subject will move the slider practically the entire possible span, all the way from an estimate near 1 to an estimate near 0, as you see here. So you see both small steps and big steps and no steps. So a general summary of the results is that you see big steps and small steps. I'll show you the joint distribution of step widths and step heights later on. One thing that will surprise some of you who know the judgments and decisions literature is that the relationship between the true probability and the median slider setting, just going across all the subjects, was essentially the identity over the entire range of probabilities. There was no ogival relationship between the objective probability and the estimated probability. It turns out that in the literature on this kind of experiment, which you're not inferring the probabilities from how people judge or the gambles they make, but you're just asking them flat out, what do you think the probability is? This is actually the usual finding. There are, going back more than 50 years, most of the, not all, but most of the experiments find the same thing that we do. As you see here, subjects generally respond quickly to changes in the probability and accurately. That is, they have a high hit rate. They seldom miss an actual change, and a low false alarm rate. They seldom say there's a change when there wasn't. And they have second thoughts, as I've already said. So let me show you a bit of the data, and then we'll get into the what do we make of all this. So as I just said, the dashed line there is the identity function, okay? I'm plotting the median estimated p, that's the circles, and the interquartile interval as a function of the true value from p's down to 0.01 to p's up to 0.99. And as you can see, if it were any closer, you'd think I was faking it. That's one striking fact. Here's another interesting datum, which is the second thought latency. So what's a second thought latency? Well, you click the I think the box has changed button, and then you see some more circles, and sometimes you say, you know, on further reflection, on further having seen more circles, I don't think it changed after all. It's the number of trials between when you said it changed and when you had a second thought. That's the second thought latency. Here's the, across all the subjects, the cumulative distribution of second thought latency. So you see that 80% of them occur within 20 circles, but there's a very long tail. The inset here is the full distribution, and you see it goes all the way out to 250 or something like that. Is that sometimes 250 circles later, the subject says, you know, I don't think there was a change. Okay, here's one of the more important and challenging aspects of the data. This is the joint distribution of step widths and step heights, right? So what's a step width? Well, you know, the slider goes along, it's not moved, that's a wide step. The more trials that go by without its moving, the wider the step. Finally, it moves, and the delta p by which it moves is the step height. Okay, so for every step, you have both a step width, which is plotted. So this is all the steps that were made in all those 100,000 trials from the 10 subjects, and this is the step width on a logarithmic scale. And you notice that, as I said before, sometimes the slider doesn't move for 320 trials. And this is the marginal distribution of step width. And here's the step height. And here's the marginal distribution of step heights. So why is this important? Well, because the obvious explanation for why the subjects don't move the slider, which I'm sure is at least 30% of the people in the audience are already muttering to themselves, well, come on, Gallistel. You know, subjects are lazy. They don't move the slider because the changes from trial to trial are usually rather small, and they just don't want to be bothered. They only move the slider when there's been a significant, after all, the position of the slider tells you what your previous estimate was, right? So even if you don't remember, you can see what your estimate was. And if you think if the estimate hasn't changed by much, you just don't bother to move the slider. You only move the slider when there's been a non-trivial change in your estimate. So it took me 6 months to convince my co-author, Peter Latham, that that couldn't possibly be the answer to this, because I kept saying, Peter, look at the joint distribution. Because this model says that there should be a hole in the step heights right here around 0 step height, because it says, look, subjects just don't make small steps. They don't like to make small steps. They're lazy. The trouble is, small steps are actually the most common. So subjects make small steps with abandon, but they don't make them on every slider setting. So we fiddled around with various models, running averages that had a threshold on the output, and there is no way that we could reproduce anything resembling this joint distribution. All right. So there's much more data behind this, but I was told to make this accessible to a general audience, so I'm sparing you some of the gory details and more elaborate arguments. So now we come to, okay, so what kind of model could explain? We're going to now take this as a literal indication of the trial-by-trial percept of the probability, and we're asking, well, what kind of model for the formation of a probability percept would have these properties? So we assume a two-stage model. This is classic modern cognitive science. This is a computational model. It's quasi-Bayesian, so it's even more model. In the first stage, the model simply asks, is my current perception serving me well, right? Is what I'm seeing consistent with what I perceive the hidden probability to be? So the really colloquial way of saying this is, it says, is my estimate broke? And I'm a Midwestern boy, and in the Midwest, we believe if it ain't broke, don't fix it. So if there's nothing wrong with your current percept, then don't bother changing it, right? That's the basic idea. And this is computationally important because an absolutely trivial computation will tell you whether there's anything wrong with your current percept. If there is something wrong, then a considerably more intricate computation is required to figure out what the problem is. So, and this is quite a general truth, that it's easier to figure out you have a problem, you know, Houston, we've got a problem, than it is to figure out what the source of the problem is and what you should do about it. So the idea here is by just going with your current estimate, as long as it works, the brain saves itself a tremendous amount of computational challenge, because on the vast majority of trials, there's nothing wrong with your current estimate, and so the slider stays where it is. Now, if, on the other hand, this very simple first stage computation indicates that there is a problem, well, then stay tuned. Then we have to decide what the problem is and what to do in order to fix it. But before I get into the details of that, I want to give you kind of the big picture about this model. The idea in this model is that your current perception is actually a kind of byproduct of a more ambitious effort, which is to construct a compact representation of the stochastic sequence that you're witnessing. That is, you're trying to extract from this non-stationary sequence the parameters, in essence, of the non-stationarity, which you can do by constructing a compact representation of it, because regularity produces compression, and compression exploits regularity. So constructing a compact representation is very intimately connected to recognizing regularities in the data. This is one of the central ideas behind minimum description length approach to model selection, for those of you familiar with that rather obscure, but I think strongly oncoming approach to model selection, both in statistics and in psychology. So on our story, this model is constructed in terms of the change points. What you're saying is, did it change? Did it change? Did it change? No, it didn't change. Oh, there it changed. Okay. By how much? Where did it change? And by how much did it change? So you always recognize a change. This is part of the interest in this problem, is that you recognize the change some trials after it happened, right? Because there's no evidence that it's happened at the moment when it happened. So your estimate of when the change occurred is never the moment at which you recognize the change. It's always, oh, well, look at that. There must have been a change back there some number of circles ago. And so you say, okay, I estimate how far back in the sequence I've been watching the change occurred. That's one number. That's the trial number. And I estimate the size of the change. That's a second number. That's only two numbers, right? Because the sequence doesn't change very often, you can represent the entire sequence of 1,000 trials with, if you have optimal parameter settings, typically on the order of 4 or 5 or sometimes 6 of these number pairs. So you've compressed 1,000 numbers into 4 or 6 number pairs, and anybody with some aesthetics would say that's a good thing to have done. And so in the process, you've constructed a model in memory of the regularities in the sequence that you've observed. And of course, the regularities are the only thing worth extracting from the sequence. They're the only, the regularities are the only thing that might help you predict the future. Whether they will actually help you predict the future or not depends on how stationary the world actually is, and we can only hope that it's sufficiently stationary to make this effort worth it. So this representation gives this way of proceeding gives you a representation of your experience that is both economical, that is, it doesn't use many physical resources if you think symbols require the use of physical resources, but it's accurate. It's actually surprisingly accurate how good the representation is for optimal parameter settings. Okay, so that's the big picture. And that's what I think is of more general interest. I'll suggest at the end of the talk that that strategy goes way beyond just estimating probabilities. So, as I've already said, in the first stage, you simply ask, you do a very simple computation. If those who want to know exactly what it is, we can easily go into that in the discussion. If there's no problem, that's it. No further computation. If there is a problem, then the challenge is to decide the most likely source of the problem. And For this, we use Bayesian model selection among three fairly obvious explanations for why what you're currently seeing doesn't tally very well with your estimate. The first explanation is that it's in the nature of this model that the estimates that you make immediately after a change are based on a very small sample, sometimes as small as 3 or 4 circles. Well, if you're estimating a Bernoulli probability from 3 or 4 circles, good luck. Your estimate is often going to be, welcome to the first idea in statistics, small sample error, right? It's going to be off because you had very little data. So that's the first possible explanation. If that's your problem, the remedy is very obvious, right? Look, you've seen quite a few more circles. So now you use all the data. You don't just use the small sample that you had originally. As you can see, this is discrete estimation. You're not continuously updating, right? The lack of steps strongly implied that whatever the process was, it wasn't a continuous update. And that, for those students of this psychological models of parameter estimation, that was a big surprise, because delta rule updating is everywhere in the literature on this. And delta rule updating, if you don't know what delta rule updating is, don't worry, it's not that important. But for those of you who do, delta rule updating, trial by trial, would produce jerks in the slider on every trial. And so would Bayesian updating. Any kind of trial by trial updating of a Bernoulli parameter estimate will produce the kind of jitter in the percept that we don't see. Okay? So the second thing, why might my estimate be lousy at the moment? Well, there may have been another change. Right? My previous estimate may have been fine, but now it's changed again. Again, the remedy, if that's what you decide is the source of the problem, is fairly obvious. You estimate where that change occurred. You add another change point to your evolving representation of the sequence. You truncate the data at the point where you estimate the change to have occurred. It's clearly madness to include data from the pre-change state in your current estimate. And you make a new estimate based only on the data that you've seen between the point where you estimate the change to have occurred and the current point, which is when you realized that it had occurred. Okay? Now, with fairly big changes, that's going to be a relatively few trials most of the time. So that's where your small sample error comes from. Right? The fact that you truncate the data, if those of you who are sort of keeping score of how this works, so that explains why the slider can move all the way from one extreme to the other. Because unlike running average trial-by-trial updating models, your current estimate is not based always on some average over the previous trials running back whatever your averaging window is, and therefore straddling the change point. Now you're truncating the data, so your new estimate is usually based on a data set that has no overlap with the data set on which the previous estimate was based, and therefore there's no constraint on how gross the difference can be between your new estimate and your old estimate. The third possibility is that, that last change point, I was suckered by a garden path in the data. I mean, that run of green circles made me think there was a change, but there really wasn't, right? And if that's the model that is most likely after you've run the Bayesian calculation, then again, it's obvious what you should do. You should extirpate that last estimate from your representation of the sequence. And now, oh wow, now you've got the data all the way back to the penultimate change point. So now you're likely to get really quite a good estimate of what the probability really is. So Why Bayesian change detection? Is Gallistel just following the fashion here, or are there actually some reasons for why are Bayesian models so popular these days? Well, change detection is inherently a description choosing problem. That is, you've got some past experience, a sequence of circles, and the brain, at least on this way of looking at things, is looking for the best, or at least the most useful, explanation of that sequence of circles. And contrasting, in the statistical sense, models or descriptions offer themselves, right? One description is there is no change point in that sequence of circles. If that's the case, you can describe the entire sequence with one number, namely your estimate of the probability of the Bernoulli process that generated that entire sequence. I hope that's clear. On the other hand, if there has been a change point, then in order to describe that sequence, you need three numbers. You need a probability before the change point, a probability after the change point, and an estimate of where the change in probabilities occurred. Right? So in a very transparent sense, the change point description of a sequence is always more complex than the non-change point description of that sequence. And one of the hooks in statistics is, therefore, it's always better. Right? No matter what the sequence, the three-symbol description of it that imputes a change point to it will always, under their pathological exceptions to this, but essentially under all real-world circumstances, the likelihood of the change point description will always be better than the non-change point description. But then an even more complicated model will be even better. You could have two change points or three. And you say, oh, wait a second, where are we going with this? Well, you could have a change point for every datum, right? In fact, if you thought describing the world was about truth, that's the right description, because it was the MATLAB random number generator that actually produced these circle sequences. And that, I'll let you in a little secret, is a deterministic process, right? If you forget to reseed the generator when you reboot MATLAB, this was a little disconcerting when we saw it in the data, you get back exactly the same sequence that you got back before, because it's actually a deterministic process, which means at the level of truth, if you believe in truth, the probability of green circle was 1 on every trial in which a green circle occurred and 0 on every trial of which. So that could be a description too. That captures the data perfectly. Right? But of course we said, no, wait a second. Whoa, yes, it captures the data perfectly, but it doesn't capture any of the regularities. It doesn't enable us to predict. And it's no more compact than the data themselves. There's something wrong here, right? Clearly, we need to strike some sort of balance between how well our model describes the data, that's the adequacy of the description, and how complicated the model is, right? And that's what brings us to Bayes, because Bayesian inference does that. So we want, this is summed up by what's sometimes called Einstein's razor. It's a version of Occam's razor. Einstein supposedly said, as usually did, this is not really what he said. What he said was not so pithy, but he supposedly said, we want our theories to be as simple as possible. That's Occam's razor, but no simpler. So the no simpler is they have to be adequate to the job, right? But otherwise, they should be as simple as possible. And Bayesian model selection, the beauty of it is that Bayesian model selection applies Einstein's razor to this kind of thing automatically and from first principles. That is, You don't have to add something in to get this. If you run the Bayesian inference, you get this for free. Those who would like an explanation of why, I'll be happy to try to do that in the discussion, but trust me, you get it for free. So Bayesian inference, and this is no small part of its attraction, perhaps not the whole part, but no small part, is that it naturally and in a mathematically principled way adjudicates between model complexity and model adequacy. And this issue is everywhere in perception. It's in a way, I like this particular case because it's so transparent. But believe me, it's everywhere in perception. All right. So why second thoughts? After all, second thoughts are sometimes thought to be a moral evil, right? Second thoughts are reviving your account of the past in order to better accord with the present, right? Well, yes, it can be abused, but it's just a fact that the future actually contains information about the current state of the world. And again, this problem makes this completely transparent, right? Early on, after there's been a change, the data are ambiguous. As more data accumulate, data you haven't seen yet bear very strongly on the question of whether there actually was a change or not. So a truly good model ought to revise its estimates in the light of further relevant data, and because you may get a better and simpler explanation for that. So one challenge, as I said, is to reproduce the joint distribution of step widths and step heights. This is now we're in the model boasting phase of this thing. And you know, it's not perfect, but it's an awful lot better. This is what we take the model and we run it on the same 100,000, we run it on every sequence seen by every subject, right? And we record, the model generates everything that the subjects generates. It has second thoughts. It has it has trial by trial estimates. It detects changes. And so we tally its statistics, and the model makes step changes. And so here we're tallying the step changes in the model and comparing the model's joint distribution of step widths and step heights when shown the same stimulus sequences that a subject were shown. And there's room for improvement, but it's a lot better than the other models we tested in generating that distribution. Again, this is slightly technical. This is for the more dedicated people in the audience about this kind of problem. You may be wondering, oh, these Bayesian models, they have priors, right? You can get away with murder in the prior, right? What are they sticking into the prior in order to get? This very issue was raised by two of my friends in discussing of another researcher who shall remain nameless. They strongly suspected this other researcher of indulging in this vice, which when you're running Bayesian models is indeed a temptation. But you'll be glad to hear that the priors in this model are Jeffreys priors. They're so-called objective priors. They're they're the least informative prior you can have. They even have a truly delicious and very intricate mathematical property. They're invariant under change of parameter. So they're chosen on purely mathematical grounds without any view whatsoever to the trying to make the model fit the data. They have another lovely property. They're so-called conjugate priors. So if you know about Bayesian models, you think, oh my God, you've got these horrible integrals. You have to do MCMC. You have to do statistical estimation of the model and so on. Well, that's all true for some models, but if you start out with a conjugate prior, you don't have to do any integration at all. The new parameter estimates are obtained by third-grade arithmetic. And when you see the update formula for a conjugate prior, you think, That's all? I just add this to that? Whoa, that's not hard. Okay, so this is computationally very simple. Even the marginal likelihoods come in closed form. That is, there's no statistical estimation of the integrals. There's just a somewhat complicated but closed form expression that gives you the marginal likelihood of all the models. And this means that prior, there are no free parameters in the prior. Because of Jeffrey's prior, the mathematical considerations dictate the so-called hyperparameters. And therefore, the entire model only has 2 free parameters. And they're the obvious parameters. If you're making decisions, you always have decision thresholds, right? And the model is making decisions. It's making 2 kinds of decisions. It's deciding, do I have a problem? That's the first stage. So you have to have a threshold on that. And then if it gets to the second stage, it's deciding, is this model better than that model? Again, so that there's a Bayes factor. And you have to have a threshold, a decision level on the Bayes factor. It's very hard for me to see how you could have a model that didn't have at least 2 free parameters. And all the predictions, the model predicts almost everything that the subjects do, lots of different aspects of their performance, and it does so with only these 2 free parameters. So it gives approximately the correct distribution of change detection latencies, for example. It gives approximately correct hit and false alarm rates. It replicates it. The model looks a lot like individual subjects. Oh, by the way, those 2 free parameters are the equivalent of alpha levels. That is, they have a statistical meaning, and therefore their values are constrained by statistical considerations, right? You're not going to set an alpha level of 10 to the minus 11 or something like that. You hope the model works with a plausible decision threshold, and indeed, the decision thresholds fall very comfortably within those that are sanctioned by ordinary usage in the experimental literature. So the model gives the identity function. It has approximately correct distribution of second thought latencies. It too sometimes says has a second thought after 100 trials. It's more accurate than the subject. Probably because the model is working with exact counts, and we're pretty sure the subjects are not working with exact counts, because we know from a lot of work that the perception of numerosity is subject to Weber's law. So the larger the numbers, the uncertainty about the true value of the number scales with the number. Now, one final result. This is a data result, but the model also shows this if you add it, if you move away from the objective priors, is that, remember, we said nothing about, we told the subjects that the box might change, but we said nothing about how often, and we said nothing about how big. Well, if you were a subject, what would you think? They said that they're going to change, and you think, well, there are probably going to be quite a few changes. As I've already told you, actually, there weren't very many changes, right? We spaced the changes out pretty widely. So 9 out of 10 subjects began by detecting more changes than they were detecting changes at a considerably higher frequency than the changes were actually occurring, which of course means that they had a fair number of false alarms. Right? The interesting thing is that all of these subjects did what this subject did. They didn't work quite as pretty as this one, but this is the cumulative record of the change detections by one subject. And a lovely property of cumulative records is that the slope is the rate. Okay? So the steeper part of this curve implies a higher rate of change detection, and the shallower part of the curve implies a lower rate of change detection. I wish Dick Herrnstein were here, because he and Skinner pioneered cumulative records, and the beauty of it is that they reveal the rate of behavior without any averaging. So they have this lovely property. Well, what you see here is that the subject, its rate, the rate at which the subject was detecting changes was slowing down, and it was moving toward the true value, the true rate at which changes actually occur, which is what you'd like to see in a really good estimator. And the model does the same thing. To make the model do that, you have to endow it with, you have to move away from the Jeffreys prior on the probability of a change, and you have to say, well, the subject started out believing that, and the other nice thing about Bayesian thing is that it's trivial to do this. That's what the prior is there for, is it's easy to say, well, the subject's distribution on what the probability of change was such and such at the start of the experiment. So it's easy to model the assumption that subjects thought the changes were going to be rather frequent at the beginning, but the model automatically updates the estimate of the probability of change in the light of its experience, as any Bayesian would tell you it ought to do. And so the great charm of Bayesian parameter estimation is that the data always dominate over the prior once you have a fair amount of data. And so the subjects, as I haven't explained because it's very technical, but the subject's estimate of the probability of a change has a strong effect on how likely they are to detect a change. Okay, so there's a feedback here. And so if they're lowering their estimate of the probability of a change, then you would expect, this isn't even faintly conscious, right? When we run ourselves, we're not even aware of doing this, right? But we think the brain of the subject is a good Bayesian brain, and it's, as the estimate of the probability of a change goes down, the probability of detecting a change goes down, as it should. Right? So the model also captures that. So general reflections. I always try to generalize way beyond what I probably should. But anyway, as I already said, we'd like to at least put out there the suggestion, and we can back this up with other perceptual phenomena, that this principle that you don't revise your perception of the current state of the world unless you detect something wrong with that perception is actually quite general. And for the reasons that appear in this model, it saves the brain an awful lot of computation. And it really makes sense that if your percept is working fine, don't change it, right? Because changing it takes considerably more computation. And computation takes energy, among other things. Also, we like this because it has second thoughts, and second thoughts are very common in perception at all levels. You think you see one thing, and then a little later you say, no, that's not that, that's something else. It has this property of using Bayesian inference to find the simplest one. So I thank you for listening, and I thank my collaborators. So I think we can just call you. Fire you right. There you are.
All right.
Yeah, okay.
Aha, here we go.
Okay. Oh, here, do you want this?
No.
Oh, okay. Well, let me turn.
All right, so very provocative. I learned something last night from Listman's discussion, which is to avoid all provocative and speculative issues from Gallistel. So I'm going to adhere to that. And in fact, I'm a visual physiologist. So what I'm going to do is I'm going to try to drag Gallistel into my world. And maybe the theme of my comments here will be that my neurons are a lot like Gallistel's model and people. And so I'm going to use a lot of the same words that Gallistel used, but I'm not going to talk about anything that he really talked about. But I'm also...
emphasizes the point that this conceptual framework is broad.
That's the hope. So I come not to bury Gallistel, but to praise him and perhaps remind him of a thing or two and you know, egg him on a little bit. So actually I'm going to skip over this because I'm going to go straight to the perception, which is the point that I think Randy was making in his last slide, which is that sensory neuron signal change in the name of what most of us think of as efficient coding. We get this from Horace Barlow going way, way back. And we can think of each neuron. This is a retinal ganglion cell, and what you can see is that when the illumination isn't changing, which is true of vast regions of images, the neuron shuts up. It doesn't fire spikes. When it fires spikes is when something in its visual field, or in its receptive field rather, is interesting. There's a contrast, there's a change. And so we can think of these neurons as having a null hypothesis, which is that there's no contrast here, nothing interesting's happening. It ain't broke, so don't fix it, right? And we can also, that leads us to an interpretation of what these spike rates are, which is something like maybe the minus the log of the probability of the image given my null hypothesis. So when I'm spiking a lot, I'm saying, ooh, something's violating my null hypothesis. And we can also see, looking at this, I don't have a laser pointer. Is there one in the house? Here, I can use this. You can also see that when something interesting happens vis-a-vis the neuron, it spikes a lot at first, and then it starts spiking more slowly, even though the stimulus is the same. But of course, so these neurons care about change over time as well as over space. This is another one of my favorite examples from the work of Hubel and Wiesel. It just makes the exact same point here in a slightly higher dimensional space, namely orientation selectivity, which is the null hypothesis for an end-stop cell is that the orientation of an edge running through my receptive field is not changing. There's nothing interesting, and we know that that is very useful for encoding things like the curvature of contours. And Fred Atneve, who was also a big information theoretic guy based on Shannon's principles, showed us that that's a very efficient way of encoding shape. So that's my first point, which is just to reinforce what Randy said, that this is a very general thing in sensory processing. And we find this not just in visual systems, but in all sensory systems, except for pain, I would say. But now we get to something that I think is a little nearer and dearer to my heart, which is that there's, and I hope, Randy's, that there's a very intimate relationship that's often underappreciated in my world between learning and perception. Now, this is a schema from an old paper by Horace Barlow, where this box here, this compare and remove matches, is the kind of things I was talking about earlier, the if it ain't broke, don't fix it. And what we're comparing these to is our current model, right? Is our current model adequate? Well, if it is, don't change anything. But if it is different, right, if there is a change, then we've learned, then there's a potential to learn something, right? There's new information out there, and we should incorporate this into our model of the current schema and also into our stored knowledge of what the environment's like. And this, I think, is where things start to get interesting. I think Randy would agree with me because I got this quote from a recent review of his, which says, I think, essentially what I've just been saying, that there's really no distinction between the two when you get down to the brass tacks. Now, this is where we have to start thinking about Bayes, I think, and probabilistic perception, because we've known, at least since the time of Helmholtz, right, that the information coming in through our senses is not adequate to define what the world is like. And so perception is an inference problem, and Bayes is a pretty good way of doing it. And this is a schema from a nice review paper by Joseph Feiser, who was at Brandeis when he wrote this. And the idea is simply this: that we take the evidence that we've been that's coming in through our senses, which is here at the bottom, and we compare this. Oops, as Barlow. Oh, where's my cursor? Oh, I think I lost something. Let's go back. There we go. All right. Okay, we compare this to our stored model of the world, and we assign it to some category. If we're pretty happy with our assignment, there's not too big an error. We have a fairly high probability that this thing is a chair, but we're not certain that it's not a table, and that gives us an opportunity to learn. So in the process, what we've done is we've updated now our model, our priors here. We've changed that little blue circle, has moved a little. And the beauty of the reason that both perception and learning need probability, perception needs it because of this problem of inference. Learning needs it because if we're going to learn efficiently, we need to update our model in proportion to whether we think it's a chair or a table, and in inverse proportion to how certain we are that it's a chair or a table, right? And this goes on again and again, and when we encounter something that we're not sure of, we have to kind of update both representations, right? And this is how Bayesian inference works. Now, I notice I haven't seen in the perceptual world models that include the second guessing option, and I think that is an interesting path to pursue in these sorts of models. Absolutely. Well, these sorts of models, so that the sampling based models, you get these bistable things for free, but yeah, that is a kind of a second guess. So maybe there is something implicit. But where things I think get even more interesting is, and this is for me one of the more interesting questions in brain science these days, is how do neurons represent probabilities, right? And this is a difficult problem. I think this is one scenario called the probabilistic population code, championed by people like Alex Pouget. And the idea here is that each neuron, I could really use a pointer. Each neuron is, yeah, I tried and it failed me. Ooh, oh, we got one. So the idea here is that each neuron, each one of these neurons has a tuning curve in this dimension. And that really the way that the brain is representing variability here, or the certainty about this stimulus, or the value of this stimulus, is by the width of this population code, right? And Pouget showed that you could do essentially probabilistic inference and Bayesian computations with these kinds of neurons or neural codes, population codes. We might think of these as sort of basis functions in a way, where each neuron is kind of representing a parameter. But there's a competing set of ideas which to most physiologists, I think, seem less intuitive, which is sampling-based code. And in this idea, each neuron is representing one of these inferred variables that the brain is trying to figure out, like the chairness of the thing that's falling on my retina. But the way that probability or uncertainty is represented here is in the neuron's spike rate over time. So each, let's say, 40 or 50 millisecond epoch represents the neuron's guess at how certain it is about that, or the value of that parameter. And if it's uncertain, we'll see a big variability in the moment-by-moment spiking. If it's not, we'll see a very small variability. Now, this seems to me less plausible, just intuitively. But actually, there's some things about it that are appealing, and I'm going to show you one result from my own lab that has made me start to think a little bit differently. In this review, they point out, Pfizer and his colleagues point out a lot of things that are different. They favor sampling-based codes. The one thing that they really point to is this problem of implementation of learning. And there's a lot of learning that can be done with sampling-based codes, and it's not clear how well probabilistic population codes can do this sort of thing, which is, I think, an important thing to remark upon. The interesting thing for me is when we get into thinking about how this might play out in the visual system. And so I'm just going to lay on you. So here's a very reduced schema of how information flows from the retina to higher orders of higher areas in the visual system. Here's the way a Bayesian might think about it. Here, I'll flip it over so that it's easier to read, which is that each one of these areas and neurons in these areas is trying to infer something. In V1, it's X1. In V2, it's X2. But it's all doing this. It's trying to calculate the posterior based on everything it knows from every source of information, including the retina, which is X0, but also including things like feedback from these other areas. And the question comes up, what would happen if we could clamp one of these areas at a fixed probability? And what I'm going to tell you is an experiment where we clamped this box here basically at 0. Right? We cooled this area and inactivated it while a monkey's doing a task. And we're going to ask what happens when we clamp this representation. And now the interesting thing, and this insight came to us from Ralph Hefner, who's a mathematician who's been a visiting fellow in my lab for the past few months, that these two different coding schemes make very different predictions about how uncertainty or variability should change when we do this. In the case of the probabilistic population code, what we're essentially doing is clamping that population at a flat prior, or at a flat, right? A uniform distribution, which should increase the amount of variability we see at the next level, right? Whereas when we do this in a sampling-based scheme, what we're essentially doing is clamping it at a delta function of certainty, right? There's no variability in any of the neurons' output over time in this when we do this. And this should lead to less variability. And what we find, and by we I mean Alex Smolyanskaya, who did the experiment, along with Camille Gomez-Laberge, who's helped with the analysis and some modeling that we're doing that I'm not going to talk about in collaboration with Gabriel Kreiman's lab, is we clamped V2, and we recorded first from this area MT. It doesn't matter what it does. We're just going to look at how its variability changed, and the answer is up here. We see a big decrease in variability, both in the trial-to-trial variability of the neurons. So if we use an identical stimulus with an identical task, the neurons will change their spike rate from trial to trial somewhat. When the brain is in its normal state, the Fano factor, which is a measure of this variability of trial to trial, starts out high. When the stimulus comes on, it goes down. Everyone sees this everywhere in the brain. But when we clamp this area, the probability or the variability goes way down. And it's not just the trial to trial of variability, it's the spiking variability of the individual neurons. So this is just one example neuron. And you can see really with your eye that the neuron spiking becomes much more regular when we've clamped this area. So that's my attempt to kind of maybe nudge Gallistel to think and provoke about this. Oh, the other thing is that we see the exact same thing in V1. So even though this clamping of V2 has opposite changes on the overall spike rate of neurons in MT and V1, we see the exact same changes in the variability of the neurons, which we think is also very interesting. All right. So I've rambled on for about 10 minutes, which is what I was told to ramble on for. So I'm going to summarize. Gallistel is right, today anyway. I think yesterday's talk was a bunch of hot air puffs, but you know, I found it very aversive. We can talk about that if we like. Sensory neurons also represent change points. Learning and perception are intimately linked. And finally, I think this question of how neurons represent probabilities is a very important and interesting one. So I guess we'll open the floor now for questions, or if Randy wants to make any comments on my comments, or if there are questions for either me or Randy, we're happy to entertain them. Wow.
Tell them about the rats. Tell them about the rats and probabilities.
I'm sorry, what?
Tell them about the rats and how rats.
Oh, okay. Quickly.
You need the...
Yeah. You can hear me? Yeah, okay.
You can use this too, Randy.
Yeah, okay. Actually, so it was the mice, at least the more recent work was with the mice. The mice are performing in a timing task in which they I didn't come prepared.
You stumped him.
It's the task. I'm trying to think of an economical way of describing the task. At the beginning of a trial, the mouse knows that if it does things right, it will get a pellet either from the hopper here or from the hopper there. But it doesn't. But on any given trial, only one of the hoppers is going to deliver a pellet, and the mouse has no way of knowing which is the right hopper. But if it's this hopper, it will deliver the pellet at the end of 4 seconds. Right? So the rational thing to do is at the beginning of the trial, go to this hopper and poke there and see whether you get a pellet at the end of 4 seconds. If you don't get a pellet there at the end of 4 seconds, then that means that on this trial, the other hopper is the one that's going to pay off, and you should now leave the first hopper and go over to this other hopper where you will get your pellet at the end of 8 seconds. These parameters have been chosen so that they're within the timing capability of the mouse, but not perfectly. That is, the mouse here, of course, just as we would, has some uncertainty about whether 4 seconds have really gone by. And so, just like the neurons show variability, the mouse shows variability in when it actually switches on the long trials. Sometimes it switches prematurely so that it leaves the short side before 4 seconds are up. If it's a long trial, that doesn't cost it anything, because the only rule is on a long trial, your first poke after 8 seconds has to be on the long hopper, and it doesn't matter how soon you arrive there. But if it's a short trial and you leave too soon, so your first poke is on the long hopper, then you lose your pellet. Right? Similarly, on a long trial, the only rule is that the first poke has to be on the long side, right? So if you stay if you stay too long on the short side so that your first poke is on the short hopper and it's a long trial, then again you lose your pellet. All right. So on this task, the mice are getting their pellets on 95 out of every 100 trials, right? I mean, we deliberately chosen this, so they're doing very well. But they're not perfect. Sometimes they lose their pellet. They leave too soon, they leave too late. So now, what if we change the probabilities of a long or short trial? Well, any decision scientist will say, oh, well, wait a second. If it's more likely to be a short trial than a long trial, then I should shift my distribution of leaving times away from the short side, right? Because I'm more likely to lose a pellet by leaving prematurely on a short trial, which are the most common kind, than by arriving too late on a long trial. Conversely, if it's more likely to be a long trial than a short trial, I should shift my distribution in the other direction. And the mice do that. We ran humans in the exact same task. You can't tell the human data from the mouse data. They're equally good at moving their distribution to the approximately optimal point. The question is, do they discover this by trial and error, or do they actually compute where it is they ought to be? Well, you can do that by looking at how abruptly they switch, because if it's by trial and error, it should be a long, drawn-out process, where if they're actually just saying, well, let's say it looks like that 9 out of the last 10 trials have been short trials, I guess we're in a new regime here, and then you should see, first of all, it shouldn't take very many trials, and second of all, you should see a step change in the distribution. Now, the statistical challenge of showing that you've had a step change in a distribution is considerable, but it can be approached with Bayesian methods. And what the data show, and actually it's painfully obvious just looking at the raw data, but of course you have to do elaborate statistics to show that what you can see in the raw data is actually true. The animals make step changes. They make short latency step changes in their distribution of switch latency. Very often, they change the distribution of switch latencies before they've lost a single pellet. So it's not reinforcement learning. It's probability estimation, and that's and you know, they're doing risk calculation, quite sophisticated risk calculations. It's not a trivial calculation to figure out what the optimal position is. So.
Variability do you see in that from individual to individual? Is there such a thing as a probability perception deficit?
You know, we didn't look at all carefully at the individual differences. Again, psychophysicists tend to regard individual differences as just annoying. The individual differences are not striking. Everything I talked about was seen in every subject. Not just in our experiment, but in Robinson's experiment. Robinson himself comments on this. He says there's striking consistency from one subject to the next. I don't mean to leave you with the impression, however, that there were no individual differences. Some people were closer than others. I didn't go into the whole issue of how you measure how close they are, which is actually itself for those of us who like this kind of issue, because if you think about it for a moment, you clearly don't want to compare to the true probability. The subjects, they don't. Only we know, only God knows what the true probability is, right? The subjects only know what the proportion they've seen is, and that departs for stochastic reasons from the true probability. So one of the reasons I think so few people have done this experiment is when they thought about doing it, they said, how would it, I mean, this is a stimulus and, how well is the subject doing? And you want to compute it trial by trial, but what do you think the stimulus is, right? I mean, what should have been the slider setting on a given trial, right? For that, you needed an ideal Bayesian probability tracker, and those have come along, but only very recently. Peter Latham, my collaborator, also Alex Pouget's collaborator, supplied that model. It's essentially the same as the Adamson-Mackay model, but that was only published in 2006 or 2007. So we used that as our gold standard of so that's where the ideal observer would have put the slider. And in that example I showed you where it looked like the subject wasn't doing so well, the ideal observer is bouncing around too, because there are runs in the data at that point. So that was an interesting aspect of it.
The model does better than the subjects, and I wonder, you said that's because the model has an exact track record of all of the exact instances, and the subjects only have an approximate representation. I just wonder if you can check that, or if you have checked that, because I think that predicts that right after a change point, the difference between the performance of the model and the subject should be very small. And as you get farther and farther away from the change point, as there's more to keep track of, right, the subject should get less accurate.
That's a really interesting point. We haven't done that. And I think you're right. I share your intuition. I should do that. these data were completed in 2009. I've been analyzing these data off and on ever since. I'm so sick of these data. But they're publicly available, okay? And I do that as a matter of principle because I'm sure there are other things that could be extracted from these data. But it's a very good point, and maybe if I've got some time one day, I'll go back
and the difference should grow with the wave refraction.
I'm sorry?
And the difference between the two should grow with the wave refraction.
Yes, and you could even measure that in individual subjects. That's a very good suggestion. Yes,
I have a related question. How are the individual events? What kind of memory does the system have? Clearly, it has to have some memory for what's come before the current event if it's going to go back. What's more, it
If it's going to detect the change. The
If it's
only
gonna
way you
detect
can
the change.
detect the change is
Right.
to compare the present.
Plus, if it can change its mind later, it not only has to remember, it has to have something more than a uniform memory for what came before. Now, one way of understanding what you said about the difference between the model and the people, one construal would be the memory for the individual events is perfect. But when you then try to summarize over segments of it, to figure out how many green things were there, the model can count them exactly and the people have to approximate them. But is it realistic? I mean, how do you think of the input of the original events
I think
serve as the basis for these calculations of probability?
I think the conclusion that they're keeping track of the individual events over a pretty big chunk of the sequence is almost inescapable. Certainly the model is. Because as you point out, when you have the second, so you're not only judging recent changes, you're going back and considering, and those earlier changes can be quite a long ways back.
Does that seem plausible to you? Strings of zeros and ones, we certainly wouldn't be able to recite them.
Well, this brings us, I think the brain has a huge amount of memory. Yeah, I mean, do I find it a bit surprising? Yes. But that's what the subjects do. So I like to think I go with the data. Not everyone agrees.
It's like this. It's like the Sperling experiment, right? You can.
Yes.
It's in there, and if you
Yes.
if you need it or you cue it in a certain way, you can.
That's a very good point. And
Well, in the Sperling experiments, it's only there for a little while.
oh, actually, so if you dig into the psychophysics literature, a lot of the recent things, there are astonishing instances in which people seem to have learned memory remember at some point in their life, and they certainly don't know they remember it, but you probe for various ways. They seem to remember astonishing things about the details of the thing that they've seen, going back quite a long way. I agree that it certainly doesn't accord with one's intuitions.
And you don't have to remember like the order of green, red, green, right? You just have to sort of have some sort of grain of.
Actually, for the change detection,
For the
you
change you do, yeah.
That's the
Yeah,
thing.
yeah.
You get the summary
But
statistics
again-
won't do you. You've got to know the order.
No, you could know
Yeah,
the
but-
summary statistics of the earlier one.
Yeah,
and the
yeah.
summary
Well, you could sort of-
statistics and realize that they're
You
not
could,
different.
you could sort of just remember P's at various grains or something, and then, narrow it in or something. Yeah, you're right, for
Part
the
of
change.
reason, so in another context, there's a form of model selection called minimum description length that is related to but not identical to Bayesian ones that I'm getting increasingly enthusiastic about, at least in certain contexts. And part of the idea behind that is by constantly constructing a compressed representation, you're minimizing the memory load because you're minimizing the cost of encoding the data. And again, this is another approach to the trade-off between model complexity and, the basic idea behind it is that you measure everything in bits. It's an information theoretic approach. So there's a bit cost to model complexity. The more complex your model, the more bits it takes to represent your model. Given your model, there's a bit cost to representing the data. So the more elaborate the model, the greater the cost of representing the model, but the lower the cost of representing the data. And the minimum description length, at least when it's working right, finds it's like Bayesian inference, but arrives at it finds the optimum. All right. That could be relevant in the sense that you're minimizing the description cost, but I don't think there's any getting around the fact that you have to remember the actual sequence, or at least some pretty elaborate summary statistics about it, going back a fairly long ways, not going back indefinitely, right? I mean, the nice thing about this model is eventually those old change points, they're frozen. I mean, because life goes on, right?
Change their minds, he said,
Sometimes after 300 trials, very rarely. But I agree, it's really startling, right? What was kind of startling is the model doesn't actually, we have no instances of the model going 300 trials, but the model sometimes goes more than 100 trials and changes its mind. But of course you say, well, of course the computer remembers every circle, right? What you're saying is, surely the human beings don't remember every circle. Well, I don't know. Yes.
Can you talk about how accurate or inaccurate people might be in perceiving probability in the social domain? So instead of estimating, how many green marbles there are, what percentage of people in this particular social group might possess a certain characteristic?
Well, I mean, I'd love it. Can I talk about? Do I have anything good to say? Informative to say? No. To my mind, there's no question that people estimate probability in the social domain, right? I mean, every adolescent male knows these statistics. I asked around, but she say yes. That's a calculation that most of us make, right? And we use various signs and things to adjust our estimate of the probability, right? How flirtatiously is she looking at me, and all these sorts of things, right? So do we make these estimates? For sure. Do I have anything informative to say about how we make them? I don't.
Would it be a different model than your two-state model?
I haven't thought about that question. It's getting into something that our model doesn't get into at all, conditional probability, right? Conditioned on the way she's been looking at me, what's the chance that she'd say yes, right? So you get into all these conditional probabilities. You probably know that this is in no way peculiar to humans. That is, as Darwin was already strongly aware of, that quite a lot of behavior is sexual signaling on both males and females, precisely because there's an obvious challenge here to calculating the odds that one or another course of behavior will lead to a desirable outcome. If a female rat is sexually receptive, if you grab her flanks, she will go into a presentation posture, which presumably is an outcome the male might like. If she's not sexually receptive, she'll try to bite you. So in a sex research lab, they go through every morning, opening the cage, squeezing the flank very quickly to see whether they get lordosis or not, right? Because they don't want to get bit, but they want to know where they are in the estrus cycle. And and there are behaviors that signal to the male where the female is in the estrus cycle, right? So that, and they have a very strong effect on the probability of the male's behavior. So I do think these calculations are going on in the brains of humans and rats, and even fiddler crabs. So fiddler crabs up there waving his big claw. Anyway, yes.
Maybe one last question from Fareed.
I'm asking this question as an attempt to understand what is meant by stages and synthesis of the model. So, suppose someone said, here is a different model, just a one-stage model. Right. You put, whether it's working or not, as another, the fourth hypothesis, the first hypothesis, in the second stage that you have the three functions hypothesis. So that's another hypothesis. Would you regard that as a different model? And I guess your answer is yes. And would that be a simpler model on your way of understanding simplicity?
Yes. Yes.
Too simple. Too simple.
So this is
Too
a
simple.
model.
Yes. Thank you.
It's a simpler model. So why did you choose the two-stage model over?
The data drove me to it. I mean, that's the whole idea behind it. At least that's what I'd like everyone to believe. That's what I believe myself. You may. Not everyone thinks the data drove me to my hypotheses about memory, but keep in mind the one-stage model. What our data and our model challenge are indeed the one-stage models, because almost all the models out there are one-stage models. And all the one-stage models, whether Bayesian or not, have the property that the estimate should change on every trial. But that's not what the data show. So we got a problem. And we got to explain, on the one hand, the fact that the slider often doesn't change for many trials. The data contain almost contradictory indications. The data doesn't change for many trials. That implies that you're averaging over a huge number of trials on the one-stage model. But then the slider changes by this huge amount in a single trial. That cannot happen if you're averaging over many trials. That implies that you're averaging over hardly any trials at all. And in fact, there are competing models out there that are running average models, but they're not two-stage, but they're two-process. You have one process that's estimating the volatility, one process that's estimating the parameter, and the volatility estimating process. They're both running averages, but the volatility estimating parameter is changing the learning rate in the other process, right? Because in a rapidly changing world, you shouldn't be averaging over very many trials. In a slowly changing world, you should be. And so the volatility estimate is altering the learning rate. That's exactly not our model, right? It's not, it's a one-stage, two-process model. Ours is a two-stage model.
So it's getting late, so I think we should thank Professor Gallistel for 2 wonderful lectures. All 10 minutes. You know, the
Sorry,
one,
you're
the one
coming to
thing.
dinner?
Yeah,
Oh,
absolutely.
okay, so
The